Hello, everybody. Welcome, everyone, to this important meeting on the advancements in addressing PFAS in Canadian water systems. I do want to note that this meeting is going to be recorded today and is being recorded as of right now. Following the successful kickoff event on April 27th, 2023 for the Investigating New Forever Chemicals in Canadian Water Systems project, we are delighted to present this webinar hosted by the Ontario Water Consortium in close collaboration with Professor Scott Hopkins of the University of Waterloo and Franco Berruti of Western University, who have spearheaded this initiative. Today, we delve deeper into the progress and ongoing research in the detection and treatment of PFAS in our water systems. We begin our session with a warm welcome from OWC, reiterating the critical need to tackle PFAS contamination to ensure the safety and sustainability of our water resources. PFAS, often referred to as forever chemicals, present significant challenges due to their persistence in the environment and potential health risks. The agenda for this meeting was shared with participants a week ago, but here you'll be able to see it on the screen here again for reference. I will now turn it over to Professor Scott Hopkins of the University of Waterloo to start off the presentation portion of this webinar.
Hey, thank you very much, Robert. Let me learn how to share my window again. There we go. Nice. So thank you all for taking the time out of your busy day to come and hear a little bit about our progress over the last year. So the list of participants is encouraging. There are lots of names that I recognize from last year. So thank you for staying engaged, and to the new names out there, welcome on board, and hopefully we can actually intersect in person at some time. So as Robert said, we're looking at PFAS in Canadian water systems, and this involves trying to detect and quantify these per and polyfluoroalkyl substances in water and wastewater, also looking at biosolids. But importantly, we also have a good thrust of trying to develop treatment technologies so that we can explore potential routes to getting rid of these little nasties that we might have. So I'm sure everybody knows about PFAS, but for the sake of completeness, let me remind you that PFAS are a group of man-made chemicals that include things like perfluorooctanoic acid, perfluorooctane sulfonic acid, Gen X. These are molecules that essentially have a fluorocarbon chain associated with them. And they're great in one way, in the sense that they have some really good chemical properties. They're resistant to heat, oil, stains, grease, water, pretty much you name it. And because of that, they're in all sorts of products ranging from clothing to food packaging, etc. But the issue that we run across here is that they don't break down very easily naturally in the environment. So we need to actually develop treatment processes. PFAS are pretty mobile. They can actually move through soils and drinking water very easily. And unfortunately, PFAS bioaccumulate in fish, wildlife, and humans. And part of the reason for that is many of them actually look like things that we need in our diet. So octanoic acid, for example, you would have eaten this morning, provided you did eat. And this is basically the good version of perfluorooctanoic acid. So for us trying to solve this issue, we first need to identify the problem. We know that we have PFAS in our environment. In fact, estimates are that 99.7% of humans have PFAS in their blood. And there was a study a couple of years ago that observed PFAS in precipitation on 7 continents. So it's out there. We don't necessarily know exactly where it is or in what concentrations we might find PFAS or how best to treat it. So we need to develop detection, monitoring, and treatment methods for PFAS. We need to build capacity for PFAS detection and treatment in Canadian water and wastewater treatment facilities. So our project team is a rather large one. I'm not going to read everybody's name because it'll be like an Oscar speech. So what I've done, though, is highlighted 5 names here, and these are folks that are going to tell you in more detail about some of the cool things that we've been doing over the last year and the progress that we've made. It's important for us to highlight our partners from the private sector. Much appreciation there, as well as Aqua OWC and CWWA, who have been very helpful in making contacts and basically being a communication pipeline for us. And of course, none of this could happen without our partners that are the boots on the ground in the treatment facilities for providing effluents, environmental waters, biosolids. I've put some people's names in here because they've been our key contacts for arranging sampling, but I know that there are dozens of other folks that I could easily have listed there. Okay, so just to remind you of our objectives and timeline, forgive me, I'm in the academic sector, so I think in terms of semesters per year. We have 3 semesters instead of 4 quarters. We're at the beginning of semester 3 at the University of Waterloo, so we are here nearly to the end of year 2, and we've had several goals, objectives that we've been progressing. In terms of the analytical side, we've developed our analysis methods for LC and 2-dimensional chromatographic separation methods. You'll hear more about that later. We've done some baseline analyses of water systems, pyrolysis treated spiked biosolid samples to give us an idea of our analytical methods and how we can use those moving forward. Same with treating PFAS spiked water. We're now looking at contaminated biosolids, just sort of starting in the area of VUV treatment for real samples because we're now at the point where we're developing that technology. This is the royal we. You'll hear from Hassan later about some of this. In terms of data processing, we've been really pushing forward on machine learning. We've developed some ML models that can help us determine the properties of PFAS and, in fact, generalized across many different types of chemical classes. And that ML structure we can actually now use to push forward into optimizing VUV and pyrolysis processes, but more data is really required to make hay in that space. So there's more to come there over the next couple of years as we collect data. Stream 3 and Stream 4 are our treatment sides of things. 3, we're looking at treating biosolids that are spiked and contaminated. Good progress there. In Stream 4, we're looking at spiked PFAS in water and contaminated waters. And we've recently deployed and have started testing a new pilot for treatment technology. And Hassan will tell you more about that eventually. Okay, so allow me to set the stage a little bit for what we're actually going to talk about. So we started out on this path with targeted chemical analysis. Basically, what we're doing is collecting and extracting samples, pre-concentrating so that we can improve our detection and quantification limits, and then using liquid chromatography to separate the components of these complex mixtures, thereby reducing ion suppression and improving our ability to detect and quantify. We do that quantification using mass spectrometry, where this is a chromatogram for all the separated PFAS that we've observed. And we've managed to get limits of detection that are sub-part per trillion. So nanograms per liter is the area that we can detect. So our targeted chemical analysis is great and in line and took a little bit of refinement over EPA methods. Amir is going to talk a little bit about that eventually. But it's led us to a few questions. One is, what about transformation products? When we start treating these molecules, what are we producing? Are those things better for us or worse for us than the original contaminants? What about the unknown PFAS that are in here? We're targeting about 40 of the usual suspects. There are about 10,000 other PFAS molecules that we're totally ignoring. And of course, other contaminants is actually a big question. What about things like drugs of abuse, pharmaceuticals, stuff like this that might be in our water? It turns out that our methodology actually translates to that as well. So here's just an example of some data that I think a lot of you will have seen already since we sent some reports out earlier in the year. This is data from a water treatment plant. I've left it as water treatment plant. Nobody's getting doxed here. We're not calling anybody out for any particular reason. We're all on the same team and we're trying to sort through these things together. So also note that we have this part per trillion concentration that we're looking at, the nanograms per liter. So in general, relatively low PFAS concentrations in water treatment plants, a little bit higher in wastewater plants. We have time series data. We're taking samples over the course of multiple weeks at influent and effluent so that we can see what's happening through the plant. We have limits of quantitation that we can report. These are about 3 times our limit of detection. So that's what the red dashed line along here is. Anything below that red dashed line, we can only say we've detected. We can't tell you how much is actually there. But in general, what we're seeing is PFAS in equals PFAS out. Whatever's going through is not actually being treated very well. And in fact, in some cases, like PFOA, there's a little bit of an increase of the concentration in the effluent, and in PFOSA, there's actually a substantial increase in the effluent, which is curious, we'll say. So something to be investigated. Okay, so what's next? Immediately following, you're going to hear some presentations from a lot of learned people. Amir is going to tell us about challenges of targeted and non-targeted PFAS analyses. Remember, we're targeting 41, but there are 10,000 that we could be looking for. Chris is going to tell us about a question mark, a new method to identify unknown PFAS. Franco will tell us about the thermal treatment process and the challenges in going from pilot to commercial scale. Josh will talk about destroying PFAS in biosolids and capturing the fluorine that's being produced. And Hassan is combining membrane filtration and VUV treatment for trying to get rid of these nasty contaminants. So over the next year, you'll be hearing from us. We're going to follow up with municipalities for new rounds of sampling. Some of that has already begun. We're further developing our treatment technologies and refining some non-targeted analysis with data. We'll be able to push forward with our ML models. So a quick nod to the people that are paying the bills for all of this great work. And thanks again for all of your help and support over the last year. And I will now turn it over to the next people in line.
Okay. Can everyone hear me well? Yeah,
okay. I can hear you.
All right. Thank you. Okay. Hello, everyone. My name is Emil Ezraich, and I'll be talking more about analytical workflow development for the analysis of PFAS in aqueous and solid samples. So first, I would go to a couple of acknowledgements that first goes to all the project partner municipalities that provided samples, and it needs to be emphasized that we couldn't do any method development on these complex samples. We didn't have some to work with because making methods on clean water is quite easy. Complex samples require a bit more work to be done. And I just want to acknowledge a couple of people from the University of Waterloo team that helped with the sample preparation that include Arthur, Shannon, Dawson, Karis, and Anish, a variety of graduate undergrad students, along with Scott and Larry, who have been guiding us. Also, acknowledgements go to Richard and Val from University of Waterloo mass spectrometry facility, who helped us facilitate running samples on their high-resolution mass spectrometry instruments, and also University of Waterloo Western Ontario team, who helped us provide samples and some discussion on biosolid and the other like VUV treatments, aqueous samples. So I will now go into a bit overview of what has been done in terms of sample preparation and LCMS analysis. So current gold standard for PFAS analysis is essentially a protocol outlined in the EPA 1633 method or something alike. However, adopting those methods from one lab to another usually requires a couple of changes, and we have identified a couple of things that we could slightly improve. One of them is using these specific cartridges from Phenomenex. The reason why is that they have a carbon inside the cartridge, which allows integrated carbon cleanup. Currently, carbon cleanup is supposed to be done in the, sequentially after solid phase extraction, and that just increases the time. And by having carbon inside the cartridge, we can reduce. We can simplify the whole sample prep protocol, and we have a less error-prone cleanup, just because we have less steps and things to transfer from one vial to another. Another one is just simply the chromatographic conditions that we have been slightly optimizing and tuning so we can separate some of the PFAS isomers. So I have here perfluorooctane sulfonic acid. The largest peak over here is from the linear form, while all these other peaks are coming from a variation of different isomers, either a slightly shorter chain with one methyl group or more short chain with two methyl groups or so. There are many isomers present under these peaks, but we can at least separate some, and this is essentially what many others are settling and what's acceptable currently. And the next is in terms of analytes that we have changed or modified from the EPA method. So because we initially, when we were buying analytical standards for these PFAS, we were looking to have more PFAS that we could cover. But there was a manufacturer error which ended up removing the perfluorooctane, the canoic acid. So in the end, we end up not really getting more, but down the line, we do plan to include it back into the study. Basically, I'd say, yeah, we can just include it back into the study. Anyway, I just want to kind of emphasize that for all sample analysis, all analyte and extract internal standards satisfy recovery ranges, which as I go to different examples, I will not talk about that, but recovery ranges are satisfied according to the EPA method. So I would like to have an overview of all the sample concentrations that we pulled from all the water treatment plant samples. And here I represented them in error bar plots with water influence and effluence and instrumental LOQs. And as Scott said, PFAS in, PFAS out, for many of them, we have very similar concentrations of influence and effluence, with PFOSA being one that is slightly skewed more towards effluence, something that we are interested in looking at. And so I've here highlighted, for example, for the reference, the US EPA maximum contaminant levels that are for vast majority of the samples, all the determined concentrations are below those levels. While for the Health Canada, currently the guidelines are sitting much higher. They are sitting above the chart. I'm not going to put a star on those. So the water is below those concentrations of what Health Canada seems safe. And I just want to take a moment now for you to see that like all the PFAS in water treatment plants are sitting in this range. But now when we go to the wastewater, all of them shifted much higher. So probably the reason for this is just we have either probably could come from the industry, from us using washing clothes or other things that have PFAS in. This graph also represents combined all the concentration for PFAS from the wastewater samples we have received. And as I said, we have about 10 times increase in this set of concentrations. And while over here for the PFOSA, it's more like mirroring the PFAS, of it between the influent and effluent. So that effect is not pronounced in the effluent, unlike the water samples. Now I'd like to go to the biosolids. So we have received a set of biosolid samples from municipalities, but we are waiting for them to accumulate so we can batch process them. But this is just one slide that I took from the optimization. Is basically if we compare PFAS biosolid extract and the clean PFAS standard, So this biosolid extract is spiked with PFAS, exact same concentration as a clean standard, and we just want to look something like similarity, how those concentrations get or instrument responses get translated. So we can see that biosolid extract, as we vary the concentration, we have a huge drop in the signal being recorded in comparison to the clean PFAS standard, which is just a result of the biosolid matrix being so complex and just interferes with the analysis. But this is just, and the PFOA is not recorded because the concentration in this specific biosolid samples that came from somewhere else, not from Southern Ontario, was so high that we adding more PFOA, we were exceeding the limits of linearity for our method. And for this set of samples, so and in general for biosolid samples, we are not going to change the default amount of sample, which is 0.5 grams. However, for the biochar samples, also a shout out to the Western team for utilizing the pyrolysis method, is that we can quickly remove most of those contaminants interfering with the analysis, and we have much better similarity instrument responses, while we are still working on investigating why these responses are getting enhanced when mixed with the biochar. And we can also increase the amount of sample up to 5 grams, and that gets much more sensitive analysis. So now looking at these other PFAS that could potentially be there, we are working on developing the overall workflow, but using high resolution mass spectrometer, which can give us a better fingerprint of identifying these PFAS. So as I said, the pyrolysis already removes most. We have about 99% of function when we look at those 41 PFAS. But when looking at others, we still have a set of features or compounds that could be PFAS. They're not necessarily all PFAS, but they could be hiding in here. And we are working on different workflows that are going to help us figure it out. So I'll just quickly go over some conclusions that we are currently, in regards to aqueous samples, we are accepting samples, we're analyzing, better processing, looking to compare seasonal changes for biosolids, accepting samples and waiting to batch process and analyze. Biochar, as Western send samples, we'll analyze, and for the untargeted analysis, essentially finalizing protocols and working on setting up to use another methods for confirmatory analysis, a method that Chris now is going to help us talk about, which can help us identify classes of PFAS and just provide a fingerprint for fingerprint of contact, essentially. Thank you very much. That's it from me.
Okay, can everybody hear me? Where's the laser pointer at? See my mouse? Okay, they can see my mouse, that's fine. Okay, thanks for coming, everybody, both virtually and in person. My name is Chris, and I'm a master's student in the Hopkins lab here at Waterloo. Amir has taught us about some PFAS quantitation using these well-known EPA methods, specifically the 1633 method, but I'm going to talk to you about something new where we've been combining liquid chromatography with differential mobility spectrometry to achieve a two-dimensional separation of PFAS. Just some quick acknowledgements before I get started. I'd like to thank, most importantly, Scott, Amir, and Larry for all their help, because they were very instrumental in the method development, especially when things break down. I know Larry and Amir have helped a lot with stuff like that. I'd also like to thank all the other people that have helped out with sample prep and running these different experiments along the way. So thank you all. Now, a quick overview of what I'll talk about. Basically, we have added differential mobility spectrometry, or DMS, to our traditional liquid chromatography workflows. And we've seen that this ends up improving separation of both unique PFAS and isomers. And additionally, we're able to reduce chemical noise in some of our samples. Now, we also see that PFAS families, the ones that Scott and Amir kind of alluded to, these carboxylic acids, sulfonic acids, have very distinct trends in our two-dimensional method. And we can use these LC-DMS trends for identifying emerging or unknown PFAS for which we don't have access to chemical standards. Like Scott said, there's about 10,000 potential PFAS that exist. We have standards for about 41. So we really want to start looking at things that are unknown. And I'll show that the instrument quantitation limits for this method are a bit comparable to LCMS. They're not as low, but on the same scale, roughly. So just a quick overview of liquid chromatography, because I want to really highlight the difference between this and differential mobility spectrometry. In LC, our analytes are separated in solution according to their hydrophobicity and basically their interactions with our analytical column. What we end up measuring is the retention time, or how long these compounds take to elute through our column. And there's a little animation there to show you a visual. Now, differential mobility spectrometry, or DMS, is different because it's measured in the gas phase. And basically, we introduce our ions into this cell, and we apply this oscillating waveform, which makes our ions kind of zigzag. And the intensity of this waveform kind of drives our ions off the center axis of transmission, and they'll end up hitting the walls. To adjust for this, we apply this compensation voltage, or CV, which steers our ions back on track so that they reach the mass spectrometer. Now, the CV for every ion is sort of unique, so it gives us another sort of identifier. So before I go into the actual 2D results, I'm just going to show you some results from both LC and DMS experiments. So if we look here at the total ion chromatogram, you'll note that they're a little bit messy. Not so much in the LC, but definitely in the DMS, they look very clumpy. You can't really identify what's going on there. So I've broken them down into the different kind of PFAS families. Starting off with the carboxylic acids, you'll see that the separation in both LC and DMS dimensions is pretty good. For the DMS, it gets a little worse as you go to these higher or longer chain species, but overall, it's pretty good. The sulfonic acids have a completely different story. First off, in LC, again, we do see separation of those isomers that Amir had mentioned, at least in the LC dimension. In the DMS dimension, you'll notice that sort of all of these sulfonic acids are not very well separated. And of course, we don't end up seeing any isomer peaks here. However, I want to point out that this minimal separation of sulfonic acids in the DMS might actually be useful for our 2D method, and I'll touch on that in a minute. Next up, we have our fluorotelomers, and there's not too much to say here. We only have a few of these compounds in our chemical standards, and they're pretty well separated in both dimensions, as you can see. And then we have a bunch of miscellaneous compounds, which really don't have any other members of their families, so we can't really tell you any kind of trends with them. But I'll note that a few of the ones here we see in the LC trace, we don't end up seeing in our DMS trace. And this is still under investigation why we don't see them. Now, when we combine both these methods together, we get this really nice 2D plot. And you can see that what I was alluding to before with the sulfonic acids being not well separated in our DMS actually kind of puts them in their own little space in this 2D plot here compared to the carboxylic acids or the fluorotelomers. But we do end up seeing these fairly linear trends between all of our PFAS families, be it carboxylic acid, sulfonic acid, and these fluorotelomers. And once again, we see the separation of our branched and linear chain isomers for the sulfonic acids. And before, we didn't really see any separation in the DMS, but if you kind of squint, you can see that these two are a little bit higher in terms of their CV, but we were just basically unable to see that because they weren't resolved in our DMS. And this is just an example of a branched chain isomer. It's not the specific structure shown here. So why are these trends useful and what can we do with them? Well, we want to start looking at those unknown PFAS, the one where we don't have chemical standards for. And we can do that just by looking for where things show up in our 2D plot. For instance, if we see a feature show up here, we might be able to say that this belongs to the fluorotelomer groups. Specifically, we might be able to classify this as the 7,2 FTS. We have standards for 6,2 and 8,2, but not this one that falls in between. And basically, the addition of the DMS, also with the reduction of chemical noise, gives us an extra identifier to help us with these species assignments for unknowns. So now we have things like retention time, CV, and we always have our mass spectrometer as sort of a third and fourth dimension. Now, in terms of the LOQs, and I want to point out that these are instrument LOQs, not the exact LOQs yet. I've put up the max concentration limits for drinking water listed by the EPA and Health Canada. The ones for the EPA are a little more rigorous and tight, but Health Canada is a little bit more relaxed as of now. And our average instrument LOQ is about 33 micrograms per liter. You see that's a little bit higher than both of these values. However, if we start incorporating this method with the EPA 1633 workflow, we can probably get up to 1,000 times pre-concentration of our samples, which gives our actual LOQ, our actual LOQ will end up being 0.033 micrograms per liter, which is more in line, especially with Health Canada. It's a little bit out of the range for the EPA, but this is obviously an average for all of these 41 compounds. You'll see that some of them are much lower than others. So where are we going with this work and what can we do? Well, first off, we want to actually start testing these on these complex sample matrices. As of now, we've only run these with clean water, but we want to start testing this on the biosolid waste or water and wastewater samples from the municipalities that are involved. We'll be doing this by spiking PFAS into these samples. And then we'll start testing this again with the biochar and bio-oils from UWO. Then we can start incorporating that EPA 1633 workflow for the enrichment of our samples, and then we'll do some LOQ validation on our low concentration samples. And then we'll start targeting, or I guess not really targeting, it will be non-target analysis, but we'll be using this LCDMS method for confirmation of unknown PFAS in the water samples and the bio-oil biochar samples as well. That's all I have, and I will pass it off now to Franco.
Hey, can you hear me? All right. Okay, thank you all for giving us the opportunity to share with you what we have been doing at Western. My team consists of myself and Naomi Klinghoffer as principal investigators, and we have a PhD student, Joshua Cullen, who is presenting just following my presentation, and Javier Ordonez Loza, who is a postdoc supporting this project. The objectives of the research is high temperature pyrolysis of biosolids for the destruction, reduction, and displacement of PFAS from the solids. The objective is really to produce valuable biochar that can then be utilized for soil amendment and carbon sequestration or for adsorption applications. Another objective is to apply thermal oxidation of the vapors and gases that are formed during the pyrolysis process for the destruction of the displaced PFAS that are going from the biosolids into the other phases. And thermal oxidation has the objective of totally breaking down the carbon-fluorine bond and separate the fluorine. So the next step is what to do with the fluorine, and the mineralization of the fluorine is another of the objectives of our research. And Josh is going to talk about it, but the objective is essentially to capture the fluorine with the metal oxides. Finally, one of our objectives is the optimization and the scale up of this technology so that they can be applied. And what we want to do is to be able to recommend to our collaborators potential technologies that can be implemented as a solution to this problem. I want to again acknowledge the wonderful cooperation that we have established with Scott's team, and for the characterization of the PFAS contaminations in the biosolids and in the products that we are generating. We have a number of external collaborations with Brown and Caldwell, USP Technologies, Chart Technologies, and everybody else that is participating in our project. Specifically, what I want to introduce today is the scale-up of this integrated process of pyrolysis and thermal oxidation, and I'm including also drying because, of course, when we deal with biosolids, we are dealing with wet feedstocks. Our experimental work at Western is carried out using a pilot plant, which we call the Bebe Show, and the pilot plant consists of a feedstock hopper on the right-hand side, and then we have a pyrolysis unit, which is a mechanically mixed reactor where heat is applied and the material is treated at a pyrolysis temperature of about 650 degrees C. Vapors and gases are generated and they are exiting the reactor from the top, whereas the biosolids are extracted from the biochar is extracted from the bottom. Vapors and gases are then either analyzed as is to determine what happens prior to thermal oxidation or directed to a thermal oxidizer where they are treated at a much higher temperature and following then analysis of the gases, of the effluents of the thermal oxidizer. This is a picture of the equipment, and I'm just going to give you a snapshot of some of the results that we have been obtaining by trying to do not really what is a material balance, because we cannot do that, not having full capabilities of analyzing all the PFAS molecules that are there, but we are doing a material balance based on the targeted components that we can analyze with targeted analysis. And what we can see from our results is that if 141 nanomoles per hour are coming into the system in the biosolids, the result is that the biochar that is produced would contain only 0.44% of the PFAS that have been introduced. So according to what we can measure with targeted analysis, again, I have to stress that. But what we see is therefore a considerable reduction of what we are detecting. And this is positive. Following the thermal oxidizer, we can see that at the end, only 0.2% of the PFAS that have been introduced are residual in the flue gas from the thermal oxidizer. And this is also another positive thing that is extremely encouraging in terms of the overall process. And essentially what we can see that based on the targeted analysis material balance, we have an overall reduction of 99.35% in the PFAS concentrations in this through the process. This is extremely encouraging. So to summarize these major findings, between 93 and 99.9% reduction was observed through the entire system based on targeted analysis. Biochar, for example, in this experiment that I showed, accounted for only 0.44% of the and the thermal oxidizer flue gas only for 0.2% of the PFAS introduced in the system. So major reduction, and obviously the objective would be to find out what else is happening and what is the true material balance of the fluorine going through the system. What we have also seen is that shorter chain PFAS comprise the majority of the PFAS detected in flue gas of the thermal oxidizer. So this indicating the decomposition of the longer chain compounds. The fluorine can be effectively captured with metal oxide, and the following presentation by Josh is going to show what we have been doing there to basically, once we break down these molecules to the extent of having the fluorine freed up, what do we do with it? So the following up question is, considering the promising results, how can we implement this in the real scale, in this industrial scale? The way we can do this, we have envisaged this type of process where the biosolids are introduced into a dryer, and they are dried to the extent of getting to maybe 20–30% moisture content to be able to be pyrolyzed effectively. So then the drier biosolids can go into the pyrolyzer, and the pyrolyzer will produce biochar as a product, which is extracted. And as we have seen from the results from Le Bébé Show, we would expect very low concentrations, or basically almost total displacement of all the PFAS that were originally in the biosolids. And the other products would be the gas and the vapors, which we take to the bottom of the chamber, and we burn them. I mean, they are combustible, therefore they can be mixed with air, and they can be combusted. So what is happening is we are integrating the thermal oxidizer together with the pyrolysis system to accomplish two objectives. One is to generate the heat for the pyrolysis itself and make it self-sustaining from a thermal point of view. And the second is to treat thermally the materials through the thermal oxidizer for a sufficient residence time and sufficient temperatures to break down the carbon-fluorine bond, which we then can pass. We can take the hot flue gases and pass them through a mineralizer where we can capture the fluorine and go to the to the dryer and utilize the heat that is still available to dry the process and to dry the biosolids that are coming in. So this is an idea, but in fact we have proceeded and we have designed a 1.2 tons per hour unit, and we are really excited that we are moving forward towards the realization of this technology, not necessarily for biosolids at the beginning. We are going to use it for wood pyrolysis and other agricultural residues, but essentially the vision is to be able to adapt this technology very effectively with biosolids. Thank you for listening.
Okay, great. All right, good morning, everyone. My name is Josh. I'm a PhD candidate working under Dr. Berruti and Dr. Klinghoffer over here at Western. Today, I would like to present some of my thesis work, which has been focused on not only the destruction of PFAS using pyrolysis and thermal oxidation, but also the capture of synthetic fluorine. So I've seen it commonly stated that thermal treatment is recognized as one of, if not the most promising option for treating sites and effluents contaminated with PFAS. And this is because thermal treatment has demonstrated its ability to destroy PFAS carbon-carbon and carbon-fluorine bonds more effectively than other technologies. And this has typically been shown using target analysis of a system's inputs and outputs, ultimately calculating a destruction and removal efficiency for a certain group of PFAS. So when thermally treated, and depending on the operating conditions, but especially temperature, PFAS chains will fracture, forming volatile PFAS such as perfluorocarbons, perfluoroalkyl acids. And extremely high temperatures, they'll form extremely short chain terminal PFAS, as well as hydrogen fluoride, and depending on the makeup or composition of the molecules, potentially water vapor, carbon dioxide, sulfur dioxide, etc. And herein lies one of the main challenges with thermal treatment. There's limited information regarding the final structure of the fluorinated products. And more work is needed identifying, quantifying, capturing terminal PFAS, which has really been a big motivation for my project. So to start, we've been doing some pilot scale pyrolysis testing with our pilot scale system with biosolids between 500 and 700 degrees. And in coordination with the Waterloo team, they were able to do target analysis of 41 PFAS in our biosolids, biochar, and biooil. So they were able to give us concentrations of PFAS initially in the biosolids, as well as the remaining PFAS within the biochar and biooil produced at different temperatures. And for the majority of the biochar, especially at the higher temperatures, these PFAS were below the limits of quantitation or limits of detection. Now, if we convert the PFAS measured initially in the biosolids into measurable fluorine and compare this to the remaining detectable fluorine within the product streams, there is less than 1% of the initial fluorine remaining as detectable PFAS in the biochar. And looking at the bio-oil, there was roughly 25 to 17 weight percent, representing two analytes, 5,3-FTCA and 7,3-FTCA. 25 to 17 weight percent of the initial fluorine still is detectable PFAS. Now, based on this kind of analysis, similar thermal treatment studies will conclude that PFAS partially degrades and displaces PFAS from the solids, with the notion being that PFAS and PFAS byproducts are said to volatilize within the pyrolysis vapors and gases. However, with this methodology, we can show the transformation, partial degradation, and displacement of certain PFAS, yet we need a way to address and quantify the final fluorine and unaccounted for fluorine in order to improve our system's fluorine mass balance. And so I've seen two ways commonly discussed for example, with pyrolysis and combustion. The first being thermal mineralization, or the idea that we can completely convert carbon-fluorine bonds into hydrogen fluoride, with a focus of key parameters such as temperature, residence time, mixing, hydrogen availability, as well as catalysts such as alumina. The idea here is that we can separate hydrogen fluoride post-thermal oxidation from the flue gas. The second method, which I've made more so a focus for my project, is coupling thermal and metal mineralization. The idea that we can add metals, for example, such as calcium, magnesium, and aluminum, in order to make fluoride minerals. The idea being that we can destroy carbon-fluorine bonds and retain the fluorine from PFAS as a solid fluoride mineral. There's been a few studies that have shown effective destruction of PFAS using these different metals, which has given us the idea to implement metal mineralization for a pilot scale testing. And there's really two approaches that we've been looking to do this. The first is with pyrolysis, mixing metals within our biosolid feed. Again, to reiterate, the idea is that we can improve our fluorine mass balance by forming these fluoride minerals such as calcium fluoride, magnesium fluoride, and aluminum fluoride. Something interesting to note is that in the ash content of biosolids, they have high amounts of calcium and magnesium already. So this could be an unforeseen synergy with thermal treatment, for example, like pyrolysis, where we could mineralize fluorine from PFAS, and therefore it would be retained within the biochar as calcium fluoride or magnesium fluoride. So we've been looking at some ways on how to implement this, or to get a better understanding of how to implement this. And we started from with some muffle furnace tests reacting fluoropolymers and metal oxides. So we've been using fluoropolymers such as PTFE and PVDF to emulate PFAS, PTFE being a perfluorinated chain and PVDF being a polyfluorinated chain, and essentially mixing these with different metal oxides to evaluate the effectiveness of mineralization. One of the analytical techniques we've been using is SEM imaging, point ID analysis, and EDX elemental mapping. So this is a zoomed in SEM image of the product of calcium oxide and PTFE mixed and reacted at 600 degrees. The instrument is able to give us an elemental estimate of the surface, as well as we are able to do a quantitative point ID analysis of certain sections of the surface. And as you can see with all these elemental weight distribution tables, we can see high abundances of calcium and fluorine and low abundances of oxygen and carbon. So this would indicate mineralization or retention of fluorine from the PTFE on the surface of the calcium. We can also show this with elemental mapping. So these are from the same images, these same SEM images, but we have the elemental maps for calcium in pink, fluorine in blue, carbon in red, and oxygen in green. You can see the affinity of fluorine on the surface of the calcium, as well as little to no carbon remaining and much lower abundance of oxygen. So all of these would indicate mineralization or retention of the fluorine from the PTFE on the surface of the calcium after reacting it at 600 degrees. To give a quick summary of some of the SEM EDX testing we've done, we've shown mineralization of PTFE, PVDF, PFA when mixed with calcium oxide, magnesium oxide, and alumina. We've also done artificial mixtures of these metals and fluoropolymers within biosolids. And what I want to talk about next is some of the testing we did with fluoropolymers mixed with biosolids, so no addition of metals. Here are four elemental maps showing oxygen in green and calcium in pink. The top two belong to a biochar sample produced from biosolids, heated at 600 degrees. If you look at the positions of calcium and oxygen, there's no real trend between the two. There are areas with high abundances of calcium, high abundances of oxygen, areas where low abundances of calcium, high abundances of oxygen. However, if we look at biochar produced at the same conditions from biosolids, yet also mixed with PVDF, when we actually overlay the images, there are clearly areas with higher abundances or concentrations of calcium and lower abundances of oxygen. And we look at an overlay of the calcium in pink and fluorine in blue. These areas are covered in fluorine. So this would be an indicator of mineralization, for example, of the PVDF, the fluorine from the PVDF with the metals within the biosolids. This was also apparent with mixtures of biosolids and PTFE. So again, to reiterate, this would indicate that PFAS precursors and potentially PFAS can react with the metal constituents in the biosolids and remain within the biochar as fluoride minerals, in this case, calcium fluoride. We also try to do some work in quantifying mineralization using X-ray diffraction. So here is a pie chart, again, from a mixture of calcium oxide and PTFE reacted at 600 degrees, showing the elemental distribution or the composition of the product. So the instrument is able to measure that roughly 22% of the product is calcium hydroxide, 35% is calcium carbonate, 22% is calcium oxide, and 32% was calcium fluoride. And based on the known mass of PTFE we added, and therefore the known mass of fluorine, and the estimated formation of calcium fluoride, we can estimate the fluorine retention within the solid sample. And as you can see, we can see really high retentions of fluorine from PTFE and PVDF with calcium oxide higher than that, for example, when comparing with magnesium oxide. So these are some of the techniques we've been using to get a better idea of how to implement metal mineralization within our pilot scale testing. The second approach we're also interested in is implementing potentially an in situ metal oxide bed for our thermal oxidizer. Since we know some PFAS will be volatilized from the pyrolysis reactor, we are looking to treat those volatile PFAS and PFAS byproducts within our thermal oxidation system as well. Again, the idea being that we can improve our fluorine mass balance for our system with the addition of metals at pilot scale. And we would like to couple this kind of testing with PFAS sampling methods such as OTM45, target analysis of specific PFAS, as well as OTM50 analysis for potential volatile products from PFAS following thermal treatment. So I would like my project to culminate into pilot scale testing with the addition of metals with the methodologies proposed for PFAS sampling. Thank you for your time. That was a conclusion for my presentation.
Hello, everyone. Can you hear me? Okay. I'm Ehsan, PhD student at Western, and today I will introduce our hybrid treatment pilot for removing PFAS from water and from contaminated water sources. But before that, I would like to mention some of our incentives and challenges in PFAS treatment of PFAS from water right now that we had it in the first place that we started this project. The first of the challenge that one of the reasons that we are gathering here today is that the conventional methods that we have for water treatment either insufficient or useless to removing PFAS from the water. And also, based on the regulations, right now industries try to shifting from the well-known long-chain PFAS towards the short-chain PFAS that might severe the situation in the following years. And another thing that is important is based on the increasing number of the studies right now for the understanding how this PFAS affect the human environment. It's going to anticipate that in the near future, the regulations for these compounds is going to be more strict. And another important thing that I would like to mention right now is about the destroying the PFAS. As you know, these compounds made of the carbon-fluorine bonds, and in order to remove them, we have to destroy all of these strong carbon-fluorine bonds. And this is very highly, for using current technologies, is highly energy intensive and is costly. And it's not surprising for for us, because whenever we want to introduce a new technology, this is one of our main challenges. So in order to reach to our hybrid treatment idea, I want to get forward step by step from more common water treatment systems to reach to our pilot. Today, everyone knows about the effectiveness of the high pressure membranes in water treatment industry. For example, using reverse osmosis or nanofiltration membranes to remove contamination from water. But this is not so permanent solution for chemicals that are famous for forever chemical. So we just put it in another stream that is highly concentrated, and we have to deal with it somehow. And also we have some kind of a degradation processes. For example, using a photochemical degradation with UV or vacuum ultraviolet lamps. But it has its own challenge. First of all, we are going to use this, for example, ultraviolet lamps to destroy chemicals that are part per trillion in most cases. So we lose a lot of energy here. We put lots of energy to remove just a tiny amount of contamination. And also we have another problem. As I said before, this is the carbon-fluorine bonds, multiple carbon-fluorine bonds in these chemicals, and just single passing through this degradation techniques, it's not going to solve the situation. It's in most cases it's going to make the situation worse, because we may start with one single chain of PFAS, then we have two shorter ones. So we have to do the recirculation, and here is the thing that's with the recirculation, the cost of the treatment is going to be skyrocket. So we have to find another way. If you look through the articles and research nowadays, this configuration is highly suggested. For example, doing the concentration first and put the all PFAS in a one concentrated stream, then using the different degradation systems, such as I said, ultraviolet or vacuum ultraviolet. But the problem is still same, is still there. First of all, based on this configuration, we are going to save some money, but it's still we have to do this recirculation over and over. And as you can see here, we are going to stick to the batch mode, and not for it's not good for commercialization. And here is our pilot that shown up, and you can see our pilot. We put a vacuum ultraviolet on a concentrated stream and do the recirculation to the feed water tank. Our pilot has three advantages. First of all, it is using it is a hybrid treatment. We use the separation and degradation both at the same time. Secondly, we use the highly energy equipment to target our contamination in a highly concentrated stream, not using for just targeting a part per trillion in this case, maybe a little bit concentrated at the source, contaminated source that we have. And also, because we have a product as a permeate, we can add a feed to the tank and run our system in a continuous mode. And here you can see how our configuration works, and it's going to be recirculated over and over between R and VUV. And to understand the potential of our system, we ran some preliminary studies before jumping into the PFAS. And we made a cocktail of pharmaceuticals as emerging contamination that we have nowadays in our water and wastewater. So here you can see that I just put two of our graphs as an example here. The first one is for acetaminophen. We ran the system in two modes, just and also it was in a batch mode to make everything simple. The first configuration, just the separation, and the second configuration in the below, you can see we add the degradation, the VUV system, on the concentrated stream. The blue dots that is representative for the just separation, you can see the piling of the acetaminophen in the feed water tank here over time. And the orange dots shows that because of the VUV on the concentrated stream, the accumulation of acetaminophen over time on the feed tank is decreasing in comparison to the just separation. And we actually did the separation and the degradation at the same time. And also, this is another pharmaceutical that we tested, the carbamazepine. It is the same trend, as you can see. The blue dots just for separation, the orange dots the separation, and the VUV at the same time. And in this table, you can see the overall degradation our system had for this pharmaceutical cocktail. And I just want to mention that we didn't use any regulation on our parameters. It's just the preliminary studies. And if we control, for example, the temperature, the pH, and this kind of stuff, the independent parameters that our pilot had, we can manage to reach the higher efficiency. And for following months and the years, I'm going to work with my partners from the different sectors, from the universities, academic sectors, and industrial sectors to work on actual PFAS samples to reach to high efficient and very cost-effective method to removing from our water systems. Thank you so much.
Thank you very much to all of the presenters. I'm just going to share my screen again and the agenda specifically. So we're now at the Q&A and discussion section of this webinar. So if you would like to ask a question to any of the presenters, you can refer to the agenda if it's for a specific person. And you can either raise your hand to ask any questions or make any comments, or of course you could also use the chat. But this is now the time to do so. So if you have any questions, raise your hand or place it in the chat and we'll address it to the right person. Wait a few minutes for people to collect their questions. So we have a first question here by Brittany Bryans in the chat. The question is, has there been any testing done on dewatered solids from drinking water treatment plant, from a drinking water treatment plant? It's not addressed to anybody specifically. Scott, as the lead, do you want to take that question?
I can do, although Larry might be able to help with this. So we've looked at dried down biosolids, but we have not looked at dewatered solids. So there is still, I guess, some space that we can explore in that regard. Ongoing, I'll say.
It's just been the influent and effluent, but it would be interesting to see from the municipal partners at the water treatment plants, if you do want to provide some dewatered solids, that would be very interesting. Again, a part of the cycle in the water treatment process that we're be familiarized with. So that we look forward to that. Thanks, Brittany.
Again, if you would like to ask a question or make a comment, please either raise your hand or place it in the chat.
did the membranes. How much of the water?
Not sure where.
For membrane separations, we can decide that how much water that we want to collect as a permeate clean water. We have a single pass through the membrane that is going to be based on the pressure that we set on the pump and the basics. That's the depend on our membrane that we use. But in overall separation that the goal that we put on our system is a 90% recovery. It means that if our pilot has 900 liters of water tank, it means that we are going to start with 900 liters of water and end with 90 liters of concentrated water and 810 liters of clean water. It is something that we actually call it in a batch mode, but in a continuous mode, this is totally different because the water goes inside, actually has a constant concentration of the contamination, so we can run the system continuously till our membranes has a good condition. But in a batch mode, our set is 90% recovery.
All right, we have a few questions in the chat now. So the first one is for Franco. The question is, do you expect the high percentage destruction of PFAS through your biochar process to be replicated in sewage sludge incinerators? I believe you felt temperatures well over 900 Celsius were going to be necessary for that. Franco?
Unmute.
You can unmute. I don't really have enough experience with the performance of typical sewage sludge incinerators, but the research that we are doing is showing that exposure of the vapors that are and gases that contain PFAS molecules, at least whatever has been displaced from the solids, exposed to temperatures in excess of 900 degrees for a residence time of at least 2 seconds, show considerable reduction in detectable PFAS in the flue gas coming out of the oxidation process. So I expect that there could be similar performance resulting from an incineration process. In fact, I believe it has been used and tested already, as long as there is sufficient high temperature and sufficient residence time to break down these molecules to break down basically the carbon fluorine bond, which is the ultimate objective.
Thank you, Franco. We have another question here from Ron Nielsen. Is there any sense of the cost of treatment / removal / destruction at this stage? Will future work include assessment of costs?
Well, this is all under development. We are trying to develop processes that are as cost effective as possible. In fact, what I was trying to explain is on a large scale, we are aiming at making a process that is self-sustaining from a thermal standpoint. The cost will obviously be capital investments and operating costs. And if the capital expense is reasonable and the operating costs are kept at a minimum by utilizing the energy available as effectively and efficiently as possible. And if there is a value in the products, in the biochar that is produced, both intrinsic value as a soil amendment element, but also in terms of the opportunities that are growing all over the world in getting carbon offset credits by storing stable carbon in the soil, the economics of this type of processes may become quite attractive.
Thank you for that, Franco. Again, any questions, any further questions, you can place them in the chat or you can raise your hand. We'll stick around here for a couple more minutes to see if any more questions roll in, and otherwise we'll be concluding the webinar. So we'll just wait a couple more minutes to see if anybody else has any further questions.
If they think of anything.
And if you have any questions afterwards, you can always get in contact with any of the participants today and presenters. Yes, there will be a recording available. It'll be posted likely through the OWC website, but we'll release that once it's all ready. And speaking to the recording, again, the recording will be likely linked to a webinar article, which we'll plan to release next week, and will be released to the OWC newsletters. So to plug OWC here, sign up for our newsletter, and you can receive all the information you need, or you can check our website. All right. Thank you very much for all the participants, to all the presenters today, for the insightful presentations. Thank you everybody for being here. We will likely have a similar webinar of sorts a year from now to provide further updates on the project. But until then, wishing everybody a great afternoon, and thank you everyone for being here. Thank you.